Thanks a lot for the introduction. I'm Ingo Müller. I'm a last year PhD student at the Natural History Museum of Sweden and at Stockholm University, and I'm happy to present to you the last chapter, or the second chapter of my PhD thesis on the beautiful Melidectes honeyeaters. But before I get into that, I thought I'd give you a quick background into what my PhD is actually about. So in my PhD, I study the biogeography of different montane avian species from the island of New Guinea. As many researchers before me, I'm very fascinated by the vast biodiversity that is harbored in mountainous regions around the world. And in my PhD, I aim to study the mechanisms that have generated but also maintained this biodiversity. Now, when we think of the role of mountains, the more intuitive role is that of mountains as barriers when they isolate populations living in the lowlands from each other. But the scenario that I'm more interested in is of populations that live on certain elevations on a mountain that become isolated from populations living on distinct mountain ranges and get separated by bodies of water or lowlands in between. Now, when we think of mountains, we all know that the environment can change quite drastically as we move upwards in elevation. And this is especially true in the tropics, where one can move from dense tropical forests up to, for now, glaciers on the mountaintops. And populations that inhabit certain elevational bands on a mountain are also more likely to be very adapted to this specific environment, which restricts their dispersive capabilities, not only from mountaintop to mountaintop, but also into these surrounding environments at differing elevations, which makes these systems especially important to study in the context of climate change. Now, when people study biogeography in mountainous areas, most of the times people will study systems in the Andes or in the Himalayas, but I would like to make a case for the world's largest tropical island, New Guinea, which is located just north of Australia. And just to give you a scale of the size of the island, if you move from one end to the other, it's about 2,500 kilometers. New Guinea boasts very diverse habitats and a vast range of climatic variation, especially as we move into its mountainous areas. And these mountain ranges themselves are also very interesting, as we have this central mountain range that stretches almost through the entire island. But at the same time, we have more isolated mountain ranges in the peripheries of the island. New Guinea itself has also fascinated many researchers before me due to its high endemic biodiversity. I myself don't go to New Guinea for fieldwork, as the fieldwork there is very time and resource intensive. Fortunately, I have access to museum collections, thanks to which I have access to large sampling sites across the entire distribution of the species that I am studying. Working on museum samples comes with its own sets of challenges, of course, especially when working with whole genome sequences, as the DNA from historical samples is damaged and fragmented. So I have to address these issues both in the laboratory and bioinformatically. Another point that I would like to highlight is that thanks to the work from me and my colleagues, we have established a lot of bioinformatic workflows that are reproducible and that I can easily apply on different data sets that I work on during my PhD. And without further ado, I would like to present to you one of these data sets, which is on the Melidectes honeyeaters. This genus consists of 6 different species, each with different width in their distributions. And more interesting to me is, however, the elevational bands that each species inhabits that vary quite a bit between the species, but there is a lot of overlap between them. The species that I will be mainly focusing on are these 3 on the top here, that were until now phylogenetically unresolved. If we take a closer look at these, we have Melidectes befordi, which lives in the higher elevations and is found basically throughout the entire central mountain range of New Guinea. We also have Melidectes rufochrysales, which is found mainly in the eastern part, in the Papuan side of New Guinea, and it lives on medium elevations. Originally, these species were considered to be just part of the same complex. That was Meyer who originally put them into the same group, but later re-evaluations of their morphology that are also linked to preferences in environment, people have decided to split these two into distinct species. Although hybrids were and are often reported between these two species that show intermediate traits. So the questions that I wanted to address in this project was first to reevaluate the taxonomic placement of these species and if it's accurate, and then later on dive deeper into the hybridization in this system, as it has not really been formally described in which species or subspecies are actually hybridizing. But I was also interested in where exactly does the hybridization occur? Is it more at the edges of the distribution, or is it just happening whenever they meet in elevational overlaps? I wanted to address these questions using a different set of methods, ranging from phylogenetic tree reconstruction to classical population genetic statistics and reconstructing their demographic history. So the first result that I would like to show to you is the mitochondrial phylogeny. And when we compare it to the previous phylogeny, we can see that at least for these outer species, it was basically the same relationships. We see that Melidectes forsteri, the green one here, that was part of the polytomy, seems to be sister to the two other species, Rufochrysales and Befordi. But when we look inside of this group here, it's quite messy, and neither species really forms a monophyletic clade, except for one clade over here, where we can find individuals only of Melidectes Befordi. And when we take a look at where these individuals are located, we can see that this monophyletic cluster is only found in the western part of the island. So after this, I had a look at the population structure. I ran PCAs and admixture analyses, and the results here align pretty well with what I also observed in the phylogenetic tree, where the outer species form their own distinct clusters, both in the PCA and admixture, but the two hybridizing species just end up in a single cluster, and they don't really become distinct in the admixture analysis. So I redid the same analysis, just zooming on to these two species, and I did recover some sort of structure here in the PCA, but this was actually just splitting subspecies of Melidectes Befordi. And the subspecies, they are also based on geographic locations. But the two species, Befordi and Rufochrysales, still don't really become distinguishable in the PCA or in the admixture results. And to show you what exactly these groups over here are, I also plotted the admixture proportions over the distributions of the two species. And what we can see is that individuals that are fixed for one ancestry are all found in this western part of the island, where only Melidectes Befordi occurs. But as soon as we go into the overlapping area, there is significant admixture in almost all individuals. And to simplify this figure even further, I plotted the admixture proportions against the longitude. And basically what we see here is if we move from west to east, it's almost a gradual increase from one admixture proportion to from one ancestry to the other. And at this point, I started thinking, this is kind of the pattern that you would expect if you force a K of 2 onto something that is just a single species, as isolation by distance would basically result in such a pattern. So I wanted to look at how differentiated these two species actually are. So I had a look at FST. And interestingly, what I saw here was that the highest value of FST was found in a comparison of populations of the same species that are found in the extremes of the island. While when I looked at interspecific FST, the value was only slightly higher than when compared to populations from the same species that are close to each other. So I seriously started questioning, why do we think that these are distinct species? I had a look then at nucleotide diversity and heterozygosity, and there I hypothesized that if they are the same species, I would expect the heterozygosity levels to be similar or the same. And that was exactly what I observed. Here I'm just showing the heterozygosity, and the only significant difference in heterozygosity was that it was lower in populations of Melidectis bifordi that were found outside of the overlapping region. So to summarize these results, I saw extensive admixture between these two species whenever they co-occur. This admixture was so extensive that I no longer believe that these two should be considered distinct species and probably are more likely to be ecomorphs, as Müller has originally suggested. And the morphology of the individuals was absolutely not reflective of the proportion of genetic admixture, as I saw that individuals that were labeled as pure parental species could still show significant admixture. So with this, I would like to thank the army of people that have supported me throughout my PhD until now, and I would be happy to take any questions from you. Yeah, so I don't have anything on assortative mating, but one of my colleagues is actually going to look outside the genome, having a look at differences in vocalizations, but I haven't seen the results yet, but that will definitely be interesting to see.
Do you see any differences in their ecologies that might suggest that they are in fact ecomorphs?
Yeah, so what was originally said is that the main difference that was described was the black beaks, and it was described that these are found more at the edges of forests, close to rivers, and the other species was found more in dense forests, and then that it was also supposedly at higher elevations and lower ones, and then as soon as you go into the overlap, you only find hybrids, but that's absolutely not what I see in the genetics, where there is no pattern of one ancestry on higher elevations or lower ones. Mm-hmm.
Is there evidence of the formula that's in the east moving to the west, like migrating and upgrading the distribution of the other one?
So of Rufochrysales, of this blue group moving towards the west. I haven't really tested directly the direction of gene flow or migration. They're also not really considered to be migratory, but there's also the issue that for many New Guinean birds, we don't really know how migratory they are. But I would say, if anything, I would think that maybe the western populations were moving eastwards, because I don't see any admixture proportions from eastern populations in the west. But yeah, I haven't directly tested it. Mm-hmm. Sure.
Given that you have museum samples, do you have the resolution to do any sort of time analysis?
Yeah, it's something that I wish I could do, but I definitely have a quite good range of time series data, but it doesn't go, I don't have enough samples to really do it on a population level scale, so to have enough individuals from the same time frame compared to more contemporary ones. I think it's also with how basically identical the genomes are at this point. I don't think that I would have recovered a pattern over, I guess I have samples from about 100 years ago. I think this must have been going on for much longer, so I don't think I would recover much a signal there. Yep. Thanks a lot.
Okay, so good morning, everyone. I'm Maria Recuerda. I'm currently doing my postdoc with Leonardo Campagna at the Cornell Lab of Ornithology. And I'm going to talk about how the cappuccino seed eater's pangenome revealed the landscape of structural variation in this species complex. So for those who don't know the system, we are focusing on the recent radiation of the cappuccino seed eaters that has resulted in this striking variety of plumage coloration in adult males, likely driven by sexual selection. So these species have diversified in the last million years, are highly sympatric, and their genomic differentiation is very low. So our objective is to study the genomic basis behind the emergence of this diversity in plumage coloration. So previous studies have found regions of the genome associated with differences in pigment concentrations at different patches between species using genome-wide association studies. So here there are two examples where we see two peaks when we compare cinnamon to black-throated species containing pigment genes, and also one peak with another pigmentation gene when we compare cinnamon to white-throated species. So we were, after that, after performing genome-wide association studies with patches, we found 6 regions that were repeatedly associated with changes in pigment concentrations across body parts, and all of them contain melanogenesis genes. So we were wondering what was the role of structural variants, because all these analyses were done with SNP data. So as we showed in a recent published review, structural variants are usually overlooked, even though they play an important role in shaping phenotypic differences. So here there are some examples of structural variants involved in generating all these differences in plumage coloration in different species. I'm not going into the details, but we found like an insertion of a transposable element and a deletion that are associated with the white phenotypes in ducks and chickens, respectively. Also, we have in quails tandem duplications and complex rearrangements that are associated with differences in plumage coloration. And if you notice, some of the genes that are marked with a red star belong to the melanogenesis pathway and are the same genes that have been detected in the cappuccino seed eaters. So one of the main reasons why structural variants are usually overlooked is because they are difficult to characterize using the traditional short-read sequencing technologies. So several methodologies are now emerging and being developed in order to better characterize these structural variants. For instance, the pangenomic approach that was developed especially for prokaryotes and then it was expanded to plants and other organisms. So briefly, the pangenome represents the full spectrum of genetic variation within a species or population complex. And basically, in this approach, what we do is generate assemblies for different species or different individuals between populations, ideally with long reads, and then we map them to generate the pangenome that includes all the shared parts among all the different individuals that is known as the core genome, and also includes the differences between individuals, which is known as the secondary genome. So right now, the prevalent method is to use a single reference genome. And the problem is when we try to map a closely related species, if that closely related species has a structural variant, in this case an insertion, when we try to map those reads to the reference, we will lose that information because that structural variant is not in our reference. But in the pangenome, if we have several individuals, it's likely that we will have that structural variant, and therefore we won't lose that information, obtaining a more complete representation of the genetic variation. So what we did, we generated, using third generation or long-read sequencing, data for 16 individuals. Specifically, we used PacBio Hi-Fi. And then we performed the assemblies for the alternate and the primary assemblies using HiFiasm, and also the annotations for the primary assembly. And all the assemblies were very similar in terms of statistics. So always the primary assemblies were longer and more contiguous than the alternate. As we see, like the primary assembly length is around 1.14 gigabases. That is very close to the expected genome length of 1.2. And then the rest of the statistics show that the primary assemblies are way more contiguous. So we have a third of the number of scaffolds in the primary compared to the alternate. Also, we have 10 times larger N50, around 31 megabases compared to 2 megabases in the alternate. and a way smaller, like 10 times smaller L50 in the primary, with 13 scaffolds on average representing half of the genome compared to over 100 in the alternate. So we also wanted to check the completeness in terms of gene content. So we used PUSCO for that, of all the primaries, all the alternate, and all the annotations. This is the average for all of them. And as you can see, all of them are pretty complete, with a very small proportion of duplicated, fragmented, and missing single copy orthologs. We also wanted to check the synteny among all our assemblies. And as you can see in the plot, there is high synteny among all of them. Here, they're just representing the 30 longest scaffolds. So as you see, some of them don't have, like, they are not connected to anything, but it's just because it's just 30 longest scaffolds of all of the assemblies. Then we also wanted to analyze the gene content of all our assemblies and annotations. So here in this Venn diagram, we see that among our 32 haplotypes, they share over 15,000 genes, and the rest of the genes are usually shared among several species. And then if we check the curve, the cumulative curve of unique genes when we add primary assemblies sequentially, we see that we reach this plateau, suggesting that with all our assemblies, we get a very complete representation of all the genes in our species complex. Then we also perform the transposable element annotation for all the assemblies. And here you can see the average percentage of the total genome length of each element. And the black bar represents the standard deviation, so all the assemblies were very similar. The most common transposable elements are the retrotransposons, with encompassing about 11% of the genome, with LTRs, lines, and signs, being the LTRs and the lines the most common, with over 5% of the genome. And then with all these assemblies from long-read sequencing data, we built our pangenome, that right now it looks like this. It has, so we wanted to understand the contribution of structural variants to these differences in plumage coloration and the differences among the different cappuccino seed eater species. So we have this pangenome with 32 haplotypes of 16 individuals, including 7 different species, and we have more than one individual for most of the species. The length of the pangenome is around 1.5 gigabytes, with 1.14 being the core genome that is shared among 90% of the haplotypes, of the haplotypes. And then regarding the variants, we recover around 46 million variants, SNPs, sorry, that are around 80% of the genome, and only 13 million structural variants that are around 20%. And this is in line with other systems where we recover around 3-4 times more SNPs than structural variants. So this is how the pangenome looks like, at least in some regions. And we see here basically some SNPs and small indels. Here we see a T insertion. But then in the pigs and other regions, it looks more like this, which is kind of a mess and very difficult to interpret, but I just wanted to show because I think it's beautiful. And then after generating the pangenome, we leverage it to map the short read data of 142 individuals to the pangenome. And with that, we were able to recover structural variants among species. After filtering, this resulted in 125 individuals and around 32 million variants, of which, again, 80% were SNPs and around 20% were structural variants. And a very small proportion, only 0.01% of the variants were structural variants larger than 50 base pair. I put the black line, but it shouldn't be visible because it's such a small proportion. And then with all this variant calling, we perform a genome-wide association study to see the associations between all the variants and the differences in plumage coloration. So Leo yesterday showed the example of the eumelanin content in the belly. Here I'm showing the example of the eumelanin content, but in the rump. And here we see that we recover, again, some pigs containing, again, melanogenesis genes. And we also are able to see that the structural variants larger than 50 base pairs that are represented in orange are not within those peaks. So also you can see that there are some differences between the two. So for instance, in the scaffold 7, there's this peak containing the gene SLC45A2 that is only recovered with the structural variants, whereas the peak in the same scaffold with the TIRP1 gene is recovered with both structural variants, but only the outlier SNPs are actually overlapping the gene. The structural variants are close to the gene, but not overlapping it. And then also the peak in the scaffold 41 is only recovered with the SNP data. So this suggests that the only small variants and small structural variants and SNPs are involved in these differences in coloration. So then we focus on the variants within transposable elements, both SNPs and structural variants, and perform the GWAS again. And this analysis recovered 3 of the 4 peaks. And this led us to wonder if the peaks that we identified using this approach were primarily driven by the variants located within the transposable elements. So what we did to test this is basically the opposite. We removed all the variants within the transposable elements and repeated the analysis. So we were expecting not to recover the peaks, but we did recover them. So this suggests that the variants within the transposable elements are, of course, important, but in combination with other variants that are outside those elements. Like the differences when we recover one peak within transposable elements is, for instance, in the AC peak, we have around 2,300 SNPs and 150 structural variants with high FST. And of those, almost a third are within transposable elements. Whereas in the peak, of the SLC45A2 gene, we have it's only recovered with the structural variants, and then when we keep only the variants within the transposable elements, we lose it. So this is basically because in that peak there are just 13 structural variants with high FST, and of those, only 2 are within the transposable elements. So then we basically repeated the same approach with FST comparisons. Here I'm showing you the pairwise comparison between Sporophylla hypoxantha and Sporophylla iberaensis. And we observed that the peaks that we identified using SNPs and SBs are consistent. And then again, that the structural variants longer than 50 base pairs are not involved in the peaks. So then we repeated the analysis, keeping just the variants within the transposable elements, and we recover 3 of the 4 peaks again. And we see the same pattern that in the GWAS. When we remove those variants within the transposable elements, we recover all the peaks again, suggesting that it's a combination between the variants within and outside these elements that are causing these differences in coloration. So to conclude, the main take-home messages are that using the pangenomic approach instead of a single reference genome improve the characterization of genomic variation. Also that small structural variants and the variants within transposable elements are involved in the differences in the cappuccino seed eater's plumage coloration, and that performing genome-wide association studies with structural variants will enhance our understanding of how genomic variation generates all this phenotypic diversity. And to finish, I would like to thank all the Cornell Lab of Ornithology, my supervisor, Leo, all the collaborators in Argentina, and all the funding institutions, and you for your attention.
Very impressive work.
Thank you.
In your Venn diagram for gene content, it's seen that there are a few species that have up to 10 or 20 unique genes. Have you looked at those to see if you can identify them?
Yeah, we look at them. It's basically because of the thresholds that are used in this analysis, because when I check those genes are in the annotations of the other species, but like probably they are fragmented, or so it's basically because of the thresholds that we set in the analysis. Yeah.
Hi, I'm Claire. I'm just starting as an assistant professor at University of Alberta, but this is some of my postdoctoral and ongoing work from the Bernachet Lab, and in really close collaboration with Claire Moreau as well. And so a lot of my work focuses on phenotypic plasticity, which is the idea that one genotype can produce different phenotypes under different environmental situations. And so recently I've become increasingly interested over this idea that plasticity can evolve over time, either to increase plasticity in response to environmental change or to increase canalization. And so I'm interested in the mechanisms that can contribute towards this. And one possible mechanism is through DNA methylation. This is an epigenetic modification that causes heritable effects on gene expression without a change in the underlying DNA sequence. Instead, it can either increase or decrease transcription depending on the context. It can affect alternative splicing and the likelihood of exons being included in transcripts. And it's also highly sensitive to the environment. And because of this, it can often lead to phenotypic changes in response to environmental changes. And sometimes those have even been shown to correlate with fitness metrics. And so DNA methylation in vertebrate genomes is generally targeted to CG sites, which is just a cytosine and directly followed by a guanine. So when sites are methylated, they have that little group there, and when they're unmethylated, they have nothing. DNA methylation is also inherently mutagenic, and often this is because of spontaneous deamination, where basically a part of a methylated base falls off, and then if it's not caught before that cell divides, it's then incorporated as a thymine, because that sort of degenerated base resembles a thymine. This primarily causes C to T transitions, and that's the mechanism for C to T transitions. But DNA methylation has also been associated with increased CG and CA mutations as well. But the mechanisms for those are not particularly clear. It's estimated that methylated cytosine can cause between 30 to 40% of all mutations in humans, and so this is potentially a huge source of genetic variation. And so what I'm interested in is under what situations can this potentially promote evolutionary change? And so if we think about organisms entering a new environment, so something like speciation, we go from a yellow fish in a yellow environment with a certain methylation state to a shift into a blue environment where that fish turns blue. And it's actually funny because this really aligns with Sebastian's talk. If you were in the epigenetic symposium, he literally showed this. So it's kind of funny. And so this can be associated with a change in methylation state. So if we think of two species sort of diverging to fill different ecological niches, their methylation state changes. And then over a ton of generations, not over a short time period, that can potentially increase the likelihood of those bases accumulating point mutations. And so over long evolutionary time periods, we lose methylation and instead can have SNPs at those same sites. And so speciation is a particularly interesting system to study this because organisms start occupying different environments. And so one of Louis' favorite systems, probably his favorite, is whitefish species complexes. And so in North America and in Europe, there are coexisting benthic and limnetic species of whitefish. So they differ in terms of a lot of traits. As you can see, the limnetic species are really small, the benthic are really large. They differ in terms of their food sources, with the limnetic species eating zooplankton, the benthic species eating sort of larger invertebrates. The limnetic species matures much more quickly. They don't grow as much, and they have higher swimming capacity in order to sort of keep up with those limnetic, faster-moving environments. And so I wanted to take a multi-omic approach to studying speciation in whitefish, benthic, limnetic species pairs. And so we sampled 4 lakes of these species pairs, 2 lakes of lake whitefish in Maine, where they're the coexisting benthic limnetic species, and 2 lakes of European whitefish in Europe. So we have about 8 benthic and 8 limnetic fish per lake. And we generated whole genome sequencing data, whole methylome data, and transcriptional data already existed from my co-author Clément's earlier study. And so we wanted to look into sort of interactions between the epigenome, the genome, and the transcriptome. For the DNA methylation analysis, we used bisulfite sequencing. And so the genome contains both methylated and unmethylated cytosines. So when you subject this to a bisulfite conversion, the methylated cytosines are not affected. The unmethylated cytosines are converted to uracil. And so you sequence the DNA, and basically for every site, if there's a cytosine there, it means that that was a methylated cytosine. If there's a thymine, that means that it was an unmethylated cytosine and it was converted. So for every site, you go through and you count the number of Cs, the number of Ts in the alignment, and that gives you an estimate of % methylation. Statistically, we assess differential methylation by identifying differentially methylated loci, which are individual CpG sites, which again are the main site of methylation in vertebrate genomes. And so when we see a difference in percent methylation between our experimental groups at a single CpG site, that's a DML. And when it's along a sort of longer genomic region, that's a differentially methylated region. So for this analysis, it was particularly important to mask SNPs from the methylation data. And this is because we are basically counting up the number of cytosines and thymines. Our cytosine reads, if we have true methylation reads for a site, are shown in black. And so here we have 4 methylated reads for cytosines and 4 unmethylated reads, which are thymines. If we have a thymine SNP at that same site, we can't differentiate between the thymine reads that are because of that SNP and the ones that are because it's an unmethylated base. And so in this case, it goes from an estimate of 50% methylation, which is the true methylation estimate, to 40%, which doesn't seem like a big deal, but when we're talking about speciation and really highly polarized allele frequencies, it becomes an issue and it can contribute to a lot of sort of artifacts in our data. And I think even with small sample sizes, this is a big issue in methylation analysis. So we took 2 different approaches to deal with this, and it's something that Clare Venney and I discussed at length with a lot of drawings. So we masked SNPs using the methylation data itself, using the max variant frac setting in methyl-DACL, and also using an allow listing approach for the methylation data, where we used the SNP data to confirm that for the cytosine position, there was no CT SNP. For the guanine position, there was no GA SNP, which would affect sort of our methylation calling for the opposite strand. And so pretty much for every base and for every individual, we determined that we had reasonable sureness that there was no problematic SNP at those sites. And so we only included SNP or CpG sites in our methylation analysis that we are pretty confident were not an issue. In terms of differential methylation between species, we saw pretty considerable epigenomic differences in all 4 lakes. We couldn't analyze the lakes sort of in a combined approach because we used 2 different reference genomes for the 2, for North America and Europe. But as you can see pretty consistently from the top annotation, we see really clear differentiation between the benthic and limnetic species. So at the very top, there's a dark blue band and a golden band, and that's differentiation between our benthic limnetic species for every lake. And from the heat maps, we can see really clear color blocking with like blocks of yellow and blocks of blue. So we're seeing really large methylation differences between these 2 species, and each line here is a differentially methylated region. There's no sort of concordance between the plots. They're different genomic regions in the different lakes. So next, we wanted to determine if there's enrichment of SNPs at CpG sites. And so across the genome, for all of the sites that we could call SNPs for, and we had enough data, we found that about 3.8% of sites were polymorphic. Depending on lake, we found that between 10.5 to 12.3% of CpG sites were polymorphic. And so both a chi-squared and a permutation test showed that this was a highly statistically significant enrichment of SNPs at CpG sites. So we're seeing a lot more SNPs at CpG sites than just in the genome on average. So this is correlational, of course. So if it's DNA methylation that's inducing mutations at these sites, the clearest mechanism causes a C to T transition. And so if we're seeing more of those, then potentially it lends a bit more support to DNA methylation inducing mutation at these sites. And that's exactly what we found. So the CT SNPs were enriched at CpG sites between 3.0 to 3.4-fold, depending on which lake we're considering. But we still found a significant enrichment of SNP, CG and CA SNPs at CpG sites. But this was more like a 1.1 to 1.3-fold enrichment. And so for this specific SNP analysis, we also only considered the mutation or SNP enrichment at CpG sites versus other cytosine and guanine bases in the genome in order to account for the increased mutation rate at Cs and Gs in the genome. And we're still seeing a huge enrichment. And so next we wanted to determine if there were sites in the genome that were both genetically and epigenetically differentiated. And so these could potentially be sites that are undergoing epigenetically induced mutagenesis. And so in some individuals, they've already mutated away from a CG site, and in other individuals, there's still a CG site and they can still be methylated or unmethylated. So we defined these as CPG sites that had both a differentially methylated locus, so where there's differential methylation between benthic and limnetic species. So on the left here, the limnetic species has really high methylation, the benthic species has really low methylation, and there's also a highly divergent SNP where there's a T in one copy for the limnetic species, but not in the benthic species. And so when we tested for an enrichment of these sites that are both genetically and epigenetically differentiated, we found between an 8 to 66-fold enrichment of these sites compared to the expected probability. And so these are sites that are both genetically and epigenetically differentiated at the exact same base in the genome. It's possible that these sites are undergoing epigenetically induced mutagenesis, so maybe some of them have mutated away from a CPG site in some fish and not others, and so they could be sort of in the process of mutating over long evolutionary time periods. It's also possible that these are just sites that for some reason their selection towards or maintenance of variation, both genetic and epigenetic, at the same sites. We can't say from this. I'd love to do experimental evolution and actually be able to kind of show this, but that's not possible with this project. So I had grand aspirations of showing more results on the transcriptome for this, and then I got an assistant professor job, and my life is kind of burning up around me in a very good way. But and so we have very basic summary statistics. Basically, we've confirmed there are transcriptional differences between benthic limnetic species in all 4 lakes, and they're actually using exons differently. And so sometimes the benthic species are more likely to include an exon than the limnetic species. So the next steps for this are to annotate these genes, figure out what they're actually doing, what their relevance could be for whitefish speciation, and then start looking into the links between the genome, the epigenome, and the transcriptome. So things like allele-specific expression analysis to link the genome and the transcriptome, redundancy analyses to link the methylome and the transcriptome and potentially pull the genome into there, some other analyses that don't really have a name, but I'd be happy to talk about them later, and also to try to understand if methylation is actually causing differences in exon inclusion in these transcripts, and so providing sort of qualitative differences in transcriptional variation. So with that, this work supports the possibility that there are epigenetic contributions to speciation, and that epigenetic divergence could, over long evolutionary periods, be creating genetic variation that then could be selected upon to contribute to speciation. Again, this is a correlational study, so I'm not claiming that we've kind of reinvented the wheel here or anything, but it does provide support for future studies, and I'm excited about potential experimental evolution studies to look further into this. So with that, I'd like to thank everyone that provided samples for this project and our sources of funding. If you're interested in learning more, the article is available online at Genome Biology and Evolution. I believe it's open access, but if not, please just email me for a PDF if you're interested. And I have to plug, I will be looking for more graduate students probably in fall 2025. We have a lot of sort of multi-omic, epigenomic projects planned, including some collaborations with Parks Canada in Banff and Jasper National Parks, which are some of the most beautiful places in the world. So if you're interested, you can find my email address, you can find my Twitter. Please come talk to me after. Thank you.
My name is Felix. Thank you everyone for attending and just more filtering in. I'm a postdoc in the Hopkins Lab at Harvard University, where we study the green things that keep our world running. I just want to say that maybe it sounds like I'm talking about the US dollar, but definitely not. We study plants, the things that turn light into food, that reproduce at distance. They entrench themselves as the literal environment within which so many other species live. So plants have evolved all sorts of unique life histories and adaptations to achieve all these things. But despite their importance to ecology, to evolution, they're not very well studied from a genomics perspective. especially wild, non-model, non-domesticated species. So by way of example, NCBI hosts 3 times as many genomes from metazoan species as plant species, and arthropods alone outnumber plants by over 1,000 genomes. I guess Haldane is happy, maybe myself less so. So there's a gap, clearly, in our understanding of plant genomics. And this gap is often attributed to the difficulty of working with plant genomes. Compared to animals, they're often large, they're often highly repetitive, and they're prone to acquiring foreign DNA via hybridization or polyploidization. And I think it's sort of a shame that these are the problems, because plants are also, these are also the very characteristics that make plants super unique and worthy of study in the first place. And plants are also often the most convenient and only avenues for probing specific evolutionary dynamics. And so I think embracing these sort of genomic challenges is a requirement. So let me introduce you to our plant representative today. Our protagonist here is these cute little roadside flowers, the Texas annual phlox. They live, as expected, in Texas. It's a clade made up of 3 species, all annuals. And here are the ranges. So Phlox drumundii is located sort of primarily in central Texas. Phlox drumundii sister, Phlox romeriana, lives in the west. And then Phlox cuspidata, which is outgrouped to these other 2, is primarily located in the east. And you'll notice that the ranges of drumundii overlap with those of the other 2, forming 2 sort of areas of contact I've tried to highlight here, in which the respective species grow in symmetry. And these naturally occurring sympatric zones has made our phlox clade into a bit of a workhorse as a setting to test all different hypotheses and models of speciation. Now, up until recently, we had very little knowledge about the genomic landscape upon which this sort of evolutionary story was playing out. So using both long and short read sequencing, as well as optical mapping, we aim to assemble the genome of Phlox drumundii, the species from central Texas. So I'm going to make some introductions here. So first, our genome is over 6 databases long. So while annual phlox, like ourselves, is diploid, its genome is twice the size of the human genome. And while the human genome is chunked into 23 nice bite-sized pieces, our phlox genome is distributed across only 7 enormous chromosomes. So if you do the math, a single chromosome is like on the order of 1 database, which breaks SAM tools sometimes in unhelpful ways. So here I've taken these 7 chromosomes and plotted them in the sort of circular plot. And not only are they big, but the genome is also highly repetitive, maybe unsurprisingly. It consists nearly 90% of repeat transposable element sequence. And this value is on the same order as maize for context, where Ts were famously first found. And so I've colored this outer band according to repeat content in 5 megabase windows. And despite our genome being big and repetitive, we're pretty confident that this is a good genome. We've also gene annotated it using RNA sequencing from multiple tissues, and we achieve fantastic annotation coverage with over 99% of boosto genes recovered here in this inner circle in pink. That's probably hard to see, so I'm going to smush our noses here against the glass, so to speak, against chromosome 2, which, like I said, is sort of on the order of a database. And I hope this provides sort of a sense of scale of what we're working with here. And you can also maybe more clearly see certain other aspects, such as how gene density generally increases towards the ends of the chromosomes, which is another feature that has been observed previously in other plants with super repetitive genomes. And so with this genome in hand, we can return to this question I posed earlier: how does evolution play out across the genome? Specifically, we know that there are these sort of areas of contact again, and we know from previous research that I just don't have time to get into, that in one of these symmetry zones, there's considerable selection on flower color to minimize hybridization. So this sort of question remains, how much gene flow is going on here? So to answer this question, we have our shiny new genome, and against it, we aligned resequenced data from over 700 samples from all 3 annual species, as well as an outgroup, Phlox pilosa. And these samples were collected as seeds by myself, but mostly others from across this annual Phlox range over the past 5 years. And we have SNP variation data from a couple of sources. We have reduced representation as well as whole genome long-read sequencing data. And the second data set also gives us, as sort of Maria alluded to earlier, into structural variation, which I'll get to later. So we have our genome, we have our two data sets. Let's get to work. So we use the diffusion approximation method DOTI, developed by Carlos Bustamante's group. And what DOTI's good at is for a given set of polymorphism frequency data and a starting tree, fitting a model of population or species divergence while allowing for migration between the populations as well as changes in population size. So for the sake of time, I'm just going to go through our best fitting model with you all. So we begin with our starting tree, in which Romeriana and Drummondii are sister species. So let's start with time. We allow DOTI to treat time before and after the split as two separate sort of epochs or parameters. So assuming a mutation rate sort of similar to Arabidopsis, the best fitting model suggests that the clade dates back about 700,000 years and that Romeriana and Drummondii diverged very soon after the divergence of Cuspidata. Next, let's add population size to the picture, which I'm denoting here using sort of relative widths of these colored boxes. And we find that all three species have population sizes that are pretty big, in the hundreds of thousands, the largest being Drummondii, followed by then Romeriana and Cuspidata. And this sort of concords with what we understand based on sort of like the relative distribution sizes, as you can see. And also Cuspidata is highly prone to selfing compared to its two sister species, which are outcrossers, which would further lower its NE. And then we also estimate NE in the ancestor of the two sister species. And here we find DOTI picking out a huge population size, over 500,000 individuals, which is just an enormous number of plants. Then finally, we asked DOTI to infer the strength of migration between each of the three populations. So our results suggest persistent migration across all three species, even between Romeriana and Cuspidata, which at present, at least, don't share any sort of sympatric or overlap zones, at least that we know of. But these sort of present-day migration edges are only a fraction of the level of migration occurring in the past between ancestral populations. And so what we have now, I think, is this emerging picture of the species that has seen tremendous amounts of migration, both in the past and in present. Now, while these migration values might appear small, if you can read them, hopefully, they're actually scaled by the population size. And so in reality, they work out to sort of like a handful of individuals per generation, which is something we see elsewhere in the data. So both structure and PCA style clustering identifies the presence of recent hybrids in the field. And when we're out there in the field, you can also just find species that carry all the hallmarks of being hybrids. Namely, they're a little bit wilty and sad looking, and they need a little bit of love. So we can further complement this complex model, these sort of model-based results with simpler polymorphism-based sharing statistics, namely the D statistic, which uses asymmetry and incomplete lineage sorting patterns to detect introgression between populations or species or what have you. So just for us, a more positive D statistic is here indicative of introgression between Drummondii, Cuspidata. Negative, more introgression between Romeriana and Cuspidata. And then, of course, values near zero suggest little introgression in either direction. And now using SNPs called this time from our long-read sequences, we test all possible triplets involving all three annual species. And again, we find evidence for favoring admixture in both directions. So after all that, the overall takeaway here is that multiple lines of evidence are now sort of showing that from different sources of data that point to the annual flux being a young clade with permeable species boundaries. Now, everything I've told you so far involves data taken from the 10% of the genome that's super well-behaved, non-repetitive. So this is often the case for demographic analyses that, where we want accurate SNP calls, what have you. But what about this other 90% of the genome that's repetitive? There was a really sad melting smiley face here, and it's not showing. My favorite emoji. But yeah, so is there any sort of useful population genetic information that we can extract from the sort of darker regions of the genome? Can we, for example, use the diversity in the transposable elements themselves within this region? to understand this, or can we use the structural variants that these transposable elements are often sort of predicted to induce to interrogate the same sort of questions about divergence and migration? So I'll just give you a quick taste, I'm running out of time, of how we're trying to answer this question. So far we've just been focusing on the Genomics assembly, and I've mentioned all of its great repeat annotations. But in fact, we have whole genome assemblies and annotations for the whole, for all of our annuals, as well as our outgroup as well. And so we can do something straightforward, like looking at the relative amount of different transposable element categories in each genome. And indeed, when we see that when clustered, these T abundances reflect the divergence patterns of our species. Now, what about structural variants? Well, let me remind you of our results from SNP data, where these statistics showed intergression in multiple directions. But we also have our structural variants from our long-reach samples. So what happens if we do the naive thing of sort of treating them like simple polymorphisms and compute D this way? Well, then we find that our structural variants are highly correlated with our SNP results, also suggesting a complex history of gene flow across our entire clade. And we also have more complicated analyses involving sort of the particular Ts associated with particular structural variants that, again, would take a little bit too much time to go through right now. Ask me about later. So both Ts and SVs provide overall useful corroborating information, though, about Flotz's history of divergence and admixture over time. Great. So to conclude, we have our genome assembly, all 6 databases, but only 7 chromosomes. This has helped us learn that the annual Flotz are a young species experiencing persistent migration, past to present, and that transposable elements and structural variants, the genomic dark matter, they do indeed contain useful information that reflects the history of Flotz's divergence. And I really want to emphasize this last point because so often, as I said, genomic dark matter is ignored when it's in fact useful and really common in genomes of species that we do and should care about. So if we want to understand the full range of what biology has to offer, to sort of understand the verbal models that only non-model organisms can answer, I think it's crucial to sort of pour effort and tools and hypotheses into accessing and unlocking all of this. So yeah, thank you for your attention. I really have to thank all the others in my lab who have helped along the way. And I'm really just a spokesperson for this really big joint effort to assemble the genome, to gather all these resources, to call variants. So thank you to Drs. Danielle Koth, Tim Sackton, Patrick McKenzie, and Robin Hopkins especially. So thank you. I'll take questions. So nervous about time.
Thank you so much for your talk. So why do you think it is the case that genes are mostly found towards the end of the chromosomes while TEs seem to inhabit basically everything coming from the centromere?
My understanding on this is not super great, and but I think it has something to do with essentially recombination. So being able to push your genes to the ends of your chromosomes ensures that you get at least one recombination event between them without messing them up. It's a very sort of like efficient way of packaging your genes when you have a lot of transposable elements. This is something we're actually interested in, but I'd be happy to chat about later. So yeah. Thank you.